I did my high school in Tirupati. And I joined at that time, it was called Regional Research Lab. Now it is called IICT. And he asked me, what do you want to work on? I said, I want
Okay.
to work on DNA. so, so
Okay.
Dr. Bhargava looked for mammalian cells that have no detectable RNA. He was working with Francis
Okay.
Crick at that time.
Mhm.
There is no such cell as cell without RNA, mammalian cell. But anyway, he found that sperm cells don't make any RNA or protein. He asked me, "Do you want to take up this project? It's going to be a very difficult project. You may have to change it if you don't succeed." Took the sperm and incubated with radioactive uridine and radioactive phosphate. So clearly, there is some amount of RNA. Even that is not possible because DNA is in a crystalline state. After working for about 2 years, I was trying, nothing worked. And luckily for me, I suddenly saw a paper where they were trying to extract sperm protein, characterize them. I added a little bit of mercaptan, boom, the DNA came out.
Okay.
So that was a lucky break I had. So the biggest criticism at that time was this is all bacterial contamination that I am looking at. Suddenly occurred to me, maybe it is not the nucleus that's making this DNA and protein, it is the mitochondria. The question is, where are mitochondria? I went to a butcher shop and and purchased, fresh liver, sacrificed from one of these bulls, and brought it and started purifying mitochondria from bull liver. I incubated sperm RNA, I isolated sperm RNA, incubated with the bull liver, so mitochondrial DNA, and I could show they actually hybridized to the mitochondrial DNA, but not to nuclear DNA. Some of the proteins made by sperm, correspond to the molecular weights made by the, mitochondria from the liver. So we communicated that paper to Nature, and it was published as a lead article in Nature. That was a great start for my career. So from that time onwards, wherever I wrote for the application, I got admission.
Wow.
So I went to UCLA Medical School. So we interpreted this as the variable regions. There are multiple copies of multiple genes of variable genes. And we are getting faster hybridization and slower hybridization with constant region. I published a paper in PNAS. All of the papers were very heavily criticized. Like you are purifying cellular RNA. There could be other RNAs which are represented by, as a contaminant. And he had shown the plasma cells make a single band. And I did this with human cells. And of course, I found a single band in majority of them. But suddenly, in one or two cell lines, I started seeing both IgG and IgM. And then later, it became clear that when B cell develops, in the beginning it makes IgM, and then it switches the constant region to IgG or IgA. Even though we did not know that, accidentally we discovered the switch of immunoglobulin synthesis that occurs in. But one mouse developed some form of leukemia which was not T cell. Out of 300, about one. It's amazing that they decided to study this one mouse. Why this is not developed T cell leukemia? That's what essentially I found was they make very small amounts of IgM. It has picked up the ABL gene from the host. So this is the so-called Abelson leukemia virus. See, there was a moratorium in the United States for recombinant DNA technology. nobody was allowed to clone a gene in US.
Oh,
So,
okay.
so Tonigawa left US and went to Basel, Switzerland, and cloned the light chain gene and showed this variable region hybridizes to a different region than the constant region. And in plasma cells, the two come together, for which he got the Nobel Prize. So we neither us or our field leader were able to do that study in the United States because we were not allowed to. So the recombinant DNA technology came and came to me and said, Prem, you are the expert in cloning. I have dozens of these viruses, acute transforming viruses. Why don't you set up a lab? We'll give you whatever you want and clone and sequence all these genes. Sequencing was not easy at that time. So I cloned Abelson leukemia virus, and then I cloned a bunch of MYC, MYB, RAS. You ask for it there. I cloned about half dozen acute transforming viruses. I had cloned the Abelson leukemia virus gene and sequenced it and showed it's a kinase. So, see, everybody was sequencing virus genes and comparing with virus, but viruses had the same mutation that the human cancers had. I had sequenced the normal gene, and people were arguing to our papers that it's all polymorphism. It's nothing, this one mutation. It will be, you will see these vitriolic editorials saying that how stupid we are, that we are trying to mutation to transformation. So, and we showed when you transfer this exon that contains the mutation, normal gene acquires transforming activity. In those days, in vitro mutagenesis was not invented yet.
Okay.
So we had to.
Oh my God, it's so much hard work, right?
Yeah, you had to switch the clones. No PCR was invented. None of PCR came much later. If PCR had come before, there would not have been any need for cloning. People who discovered cloning got Nobel Prize, and So and this guy was willing to. He had collected lung tumors from a bunch of cancer patients, and Mariano said, "Can you get me the normal tissue from these patients?" Oh, he said, "Okay, some of them are still alive. I can get their blood, and which will have normal DNA." So he sent us a bunch of these samples, and Mariano, did DNA transfection, showed that one of the tumors transforms the NIH3T3 cells. Then I picked that up and cloned the RAS gene and sequenced it and showed it had a mutation, and his normal tissue did not have the mutation. So again, we published that in Science, and again, it made, even though it's a trivial, we knew it from day one. This is just, satisfy the criticism.
The polymorphic.
Polymorphism. Overall polymorphism theory. And that became we published that. And by then, all three families of KRAS, and the one I sequenced from lung was KRAS gene. So, and actually, ironically, will you believe it or not, it has the glycine to cysteine mutation. So now,
that's what you have discovered.
At that time, this is 35 years ago. And it is that cysteine that is now being targeted by these new RAS drugs. So it
took 35 years to target that, right?
Yeah. So it was very fortuitous that we had a lung cancer patient. And in unknown reasons, in lung cancer, 30% of RAS mutations are glycine to cysteine. So it's a question of numbers. We got it because that is the most predominant mutation. And now, people are finding out 30% of lung cancers have cysteine mutation, and these drugs can target the
Okay.
cysteine mutation.
Wow. Wow. So this is the discovery of
Of
proto-oncogenes or real
Oncology,
oncogenes.
it's an oncology.
Yes, sir.
So
Wow.
from then onwards, I moved to then I moved to Hoffman LaRoche,
Yeah.
thinking that we can develop a drug for BCR-ABL. So we
Okay.
went there. I collaborated with some of the chemists there, and actually I took two projects with me. Dr. Gallo had just then cloned the HIV gene. He wanted me to sequence it. So I told him, look, I'm leaving NIH and moving to Hoffman LaRoche I have taken up a job there in Roche Institute of Molecular Biology because I want to develop small molecule inhibitors for kinases. And he said, okay, I will give you my clone. Can you develop a vaccine for it? Because Hoffman
Okay.
LaRoche was very well known for protein expression.
Okay.
So I took his clone and I expressed the HIV envelope protein, and this is more to as a vaccine candidate. But then I told Gallo that that I cloned and expressed the HIV envelope. He said, "Oh, I will send you 100 sera. You tell me which one reacts with it." Okay,
Okay.
and we screened his thing, and there were about 40 positives and 60 negatives. I called him
Oh,
and told him,
okay.
no, these 40 are positive, these 60 are negative. He was completely shocked. It was 100% correlation with HIV infection.
Oh, okay.
So
And
that was,
okay.
we patented it as a diagnostic test, and I still get royalties for HIV diagnostic test.
Wow.
NIH.
Okay. Wow,
So,
fantastic, sir.
so
Okay.
my second project, see again, Hoffman LaRoche felt there is no market for HIV diagnostic because it's such a small disease. They did not understand it is not the disease that you are going to detect. You want to detect the virus in normal blood. Those blood banks were all contaminated with HIV donors, donors which were infected with HIV. And they missed a golden opportunity of becoming a major developer of HIV diagnostics. So Abbott essentially developed it, and they made billions of dollars. And again, with the ABL, we could identify initial leads that bind and inhibit the kinase activity. But Hoffman LaRoche again felt there is not enough market for CML because CML patients were only about 3,000 patients. So they were calculating maybe you give this drug for a month and the disease goes away, and there is, and
So how was this? This is one of the very important question that every biologist or every scientist can ask themselves. Right? Now you are thinking like an industry, right? Now before you were an academic, everything was open for you, but now suddenly industry kills two projects. One is HIV, and the other one is the oncogene project.
yeah.
And now you suddenly have to. I mean, everything now is money, right? Not
So that's the whole
excitement.
problem. They made a major mistake. Their market calculations were. I was a scientist. I had no clue how they calculate
Yes.
market size and all that.
Yeah.
They were calculating. There are maximum there can be 10,000 HIV patients, 10,000
Okay.
diagnostic kits. How much you can charge? How much you will make? It is not 10,000, it had to be millions because HIV was it spread so fast. Plus, we had to screen every normal person to whether he's HIV positive or not.
Mm.
So they were unwilling to make this radical change in calculations. It's
Okay.
not a diagnostic for sick people, it is diagnostic for normal people.
Yeah.
You know, and blood banks. Blood banks were heavily 20% of blood was contaminated. So no blood from Red Cross could be used for transfusion until it went
Okay.
through a test. Even
Yeah.
today,
Okay.
even though HIV is
Definitely.
very low in
Not
percentage,
prevalent.
every
Yeah.
blood donation is screened for HIV. That's how they missed the market. And again, in the
Okay.
able, Novartis developed 10 years later, Gleevec, 10 years
Okay.
after we first made this observation in Havel-Roche. And it became a multi-billion-dollar drug,
Yeah.
mainly because you don't give it for the patient for 1 month. The patient has to take the rest of their lives.
So these are the beginnings of chemotherapy, let's say.
Yeah, these beginnings of targeted therapy. See, they were following the model of chemotherapy. See, Taxol was there. Taxol is given 6 doses and at the 3-week intervals. And after 6 doses, the patient cannot survive. So they withdraw the drug. And if the cancer is cured, it is cured. If it is not, the person is going to progress and die. So that is the Taxol model. That's all
because
chemotherapy.
it's a very, let's say, dirty drug. Not dirty, maybe it has a lot of side effects.
You cannot take more than 6 doses. That's it. You will die. So they were following that model. The Gleevec model is totally different. You don't give once in 3 weeks. You have to take every day. And you have to take throughout the life. It's not 30 days. It is not, it's several, tens of thousands of days. And there is another, a good thing that happened, both for the patient and for the company. Because nobody is dying, the number of CML patients used to be constant. It used to be about 3,000 in US. It has become 30, 40, 50,000 because they are not dying of the disease. All these people have to keep taking the drug for the rest of their lives. So that's how it became a $3 billion drug.
Oh, wow. So many choice, opportunities are missed because of wrong assumptions, right?
This constantly missed in pharma industry.
Yeah. Wow. Okay. Great.
So
So from Roche, you moved back again to academia also.
I went, I felt very disappointed. Here are 2 major projects. I thought I'm going to win Nobel Prize, and these guys didn't even want to develop it. And what is the purpose of staying in a pharma company? So I was getting ready to move, and I actually, I went for a meeting in NIH to discuss about this vaccine project. And at that time, I met Hillary Koprowski. And Hillary Koprowski said, look, I don't have a virologist. I will give you the deputy director's job. Come and join my institute. So I took that job. And then 6 years later, I was offered the directorship of the Phelps Institute. I went there mainly because it had a very good medicinal chemistry program. So I felt I can go there and start developing experimental therapeutics program I can develop. And so that is briefly my career.
Wow. So you said you missed Nobel Prizes. So what is your comment on it? Because
I see your work for Nobel Prize, but it is lots of we were all nominated when we discovered point mutation in RAS, but nobody ever got. And I think Weinberg. I was meeting with Weinberg. I was saying, I'm still thinking we will all get Nobel Prize one day for RAS. It has to be cured first. He's still hopeful.
Yeah, but sir, you also have a lot of compounds in the clinical trials now, right? So when did the journey started? Because after Hoffman La Roche, you came back to academia, then you started to work on medicinal chemistry, or maybe
Yeah, that's
also some
when
kind of
I,
synthetic chemistry, right?
yeah.
Yeah.
So when I was in, I mean, Vista Institute is one of the best institutes I worked. NCI,
Okay.
if I rate in, and I put NCI as number one and Vista Institute as number two because it's an open institute. You can do whatever you want. You can collaborate. It's in the middle of the University of Pennsylvania campus, which itself has vast resources, and all of us had appointments, joint appointments with the Penn, and we had a lot of very fertile interactions. But what it lacked was a very good chemistry program to make
Okay.
new compounds and stuff like that.
Mhm.
And the solution was to collaborate with somebody in the chemistry department of University of Pennsylvania. So a lot of times collaborations, you don't have the kind of freedom you want. So when they offered me the directorship of the Fels Institute, it had an experimental therapeutics branch. Okay. So I went there and I starting with kinase inhibitors, RAS inhibitors. These were my two major focus. So we developed several compounds. Again, I learned a lesson there. We developed lots of compounds, and we started this company called Onconova. And I handed over all my patents to this company. What I didn't realize was a company can, especially a startup, cannot develop more than 2 or 3 compounds. So I had at least 20 different compounds which had very good anti-cancer activity, and they never developed them. And their patents have slowly over a period of time expired because after 15 years the patents expire. So that's now I'm doing 2 or 3 compounds per company and trying to so people say,
So
oh, you
it's more
already have a
strategic.
company, why do you want to have another company? I said, this is the problem. If you have one company, it has to be a multi-billion-dollar company to develop 10 compounds. Even Merck doesn't want more than 3 at a time.
But these are like huge investments, right? Like maybe $1 billion or something for 15 years. And if even one fails or 2 fails or everything fails, that's going to be a huge loss.
Yes.
Okay, great. So do you want to tell a few things about the compounds that you have developed and what is exactly
So the
where they
latest,
are now?
see, one of the compounds which we developed targets RAS, okay? And that drug is still, see, the thing with that compound was, again, the experience of a CMO, Chief Medical Officer, is very important. So again, the company Onconova made a huge mistake. This is a compound that targets RAS. And they were doing clinical trials, not only in patients with RAS mutation, but also those without RAS mutation.
Oh, okay, like P53 or something, right?
Yeah, some of the, see, they were doing with, they did initially, because the mechanism was not clear. They did
I mean, as for me, it looks like a common sense, right? But unfortunately, okay.
So they
Something
did
is targeting RAS and patient doesn't have RAS, so it doesn't work.
So, no, you have to know the background of this. So they screened a bunch of patients, different patients. It seemed to work quite well in a disease called MDS, myelodysplastic syndrome. So, and they did a phase 1 and a phase 2 trial. And it was very, myelodysplastic syndrome is a kind of is a conglomeration of diseases, all with the same phenotype. So the disease itself is classified into multiple classes based on mutations, chromosomal translocations, and all that. So the broad classification is there is actually a term, low risk and high risk,
Okay.
MDS. So the low risk MDS is not, is a very mild disease which slowly progresses to high risk MDS.
No.
And our compound was working extremely well with high risk MDS. Okay. And within that group, there are multiple mechanisms. And there was a group called, based on chromosomal mapping, they said trisomy 8, that means this it had 8 chromosome 8, and monosomy 7. Okay, they had only 1 chromosome 7. But later on, what people found was these two categories of MDS have high proportion of RAS mutation. Almost 70% of those patients have RAS mutation. So that's why they were responding to this drug extremely well. So in phase 2, they could clearly see this distinction between this. These were still classified as high-risk MDS. Okay. So when they went to phase 3, we had a collaboration with Baxter. It's a European pharmaceutical company. And they were not very happy that we are treating such a small group of cancers. They wanted to expand, the number, the type of patients that are treated. So they wanted to include all high risk, whether they had RAS mutation or not.
Oh, okay. Okay, so it was again a strategic decision.
Then it showed survival benefit, but was not statistically significant because we have diluted it with non-RAS.
Non-carriers. Non-RAS, yes.
So they are redoing this whole thing with RAS mutant cancers, especially in lung. Mount Sinai has done a study in combination with immune checkpoint inhibitors, and it is showing response. So again, the problem there is the formulation they had. See, these are all very important details in drug development. When you inject a drug, it has to stay in blood circulation for a minimum of 8 to 12 hours, for it to be effective. The formulation, it is actually controlled by, now there are various techniques of formulation. Slow release, you would have heard, slow release formulations, where a capsule releases over a period of time so that all the drug is not released one time and excreted quickly. None of those things were done with rigosertib. It was cleared from the blood within 3 to 4 hours. That was not adequate. They were giving 2 pills, one in the morning, one in the evening. But a little more work on formulation would have fetched much better results. I mean, I think even now that opportunity exists for that. And also,
so phase 3 is still going on. Phase 3 is still going on.
There is, and we developed a CDK4 inhibitor. See, one of the studies we did when I was at Fells was show when you knock out, so we were interested in understanding the role of these genes in normal development. So CDK4 gene was a very important gene. It's a cell cycle regulator. So one of my graduate students, this kid's name is Sushil Rane. He came from Bombay University, joined my lab as a graduate student. And he created a mouse where he knocked out CDK4 gene. And we were expecting this mouse will not survive. It survives fine. Okay. But it develops diabetes after some time. Okay. So nobody knew until then there is a connection between CDK4 and diabetes. It's mainly because CDK4 is very important for pancreatic beta cells for replication. So over a period of time, there is an attrition of growth, and these islet cells disappear, and that's how they develop diabetes. But at the same time, what happened was we were cancer biologists. I was less interested in diabetes. So we were trying to find out how important is CDK4 in breast cancer, for example. So we had mice which were MMTV new transgenes which develop breast cancer. And when we mated with these CDK4 knockout mice, these knockout mice, even though they have the activated oncogene, don't develop breast cancer. So we postulated CDK4 is an important target for breast cancer. And we developed one. And Onconova, was putzing around, and then Pfizer developed a CDK4 inhibitor. Eli Lilly developed a CDK4 inhibitor for breast cancer. And both those drugs were approved. And ours is still in clinical trials. It's a question of putting resources in the right drug and doing it.
Oh my God. Okay. But you never collaborated with them directly, sir? Like the big pharma companies from the beginning?
No, but see, the license was licensed, my drug was licensed out by Onconova. So they were unwilling to go anywhere else. They were not willing to give a patent.
Oh, okay.
So those type of problems. So that's my message for all entrepreneurs who are starting startup companies. Think carefully. How to do a clinical trial and in what type of patients you should do, and not be greedy about it. Oh, we want a bigger market. That's not going to. Bigger market is not going to help the success of your clinical trial.
Wow. Okay. These are the lessons when you convert yourself from a basic biologist to a translation biologist.
Yeah.
Or translation
not
scientist.
only that, you have to start learning business.
Yes.
Yeah. Like Steve Jobs and these people are very successful entrepreneurs because not only they were great technologists, but also they knew how to manage business.
Exactly.
You can tell Apple when he left, the Apple collapsed almost nothing.
Yeah. Sir, wow, I think it's been more than 1.5 hours. I'm kind of enjoying the journey, what you shared, like a chemist who had no idea of biology, then it developed curiosity, the biochemistry, and now your whole life is biology. And now coming back to chemistry with your compounds, right? Again, targeting the biology. Yeah. So, sir, you also founded a couple of journals, right? When I read through Oncogene is one of the very famous journals.
So our problem, when we started doing this oncogene work, was, to start with, when I cloned a large number of these genes. So a lot of people got these clones and sequences from me to do additional work. So, and I used to go to all these meetings and participate in a lot of discussions. And at that time, Elsevier publishers came and asked me whether I can publish, whether I will be willing to edit a book on oncogenes. So at that time, I realized there is not a comprehensive book which describes, like I said, there are all these sequences that are there. Nobody knows what is related to what. Computer technology in terms of sequence comparison and all that was not well developed. And only a few people could do it. So we needed a book which gives a comprehensive picture of each of the major oncogenes that are being worked. So I was at that time in Hoffman LaRoche. So I had two colleagues, Ann Skalka and Tom Curran. They're both virologists. Tom Curran is the person who cloned the FOS gene, who'd actually discover a FOS gene. And the three of us, put together a book called Oncology Handbook. So that was actually, at that time, at least in 80s, it was widely used in all libraries. Then, we started facing a problem. There were a very limited number of journals in the oncology field. So there were either virology journals like Journal of Virology, Annals of Virology, those type of things. And there was only one cancer journal, which was Cancer Research. It's again a AACR journal, American Association of Cancer Research. And almost all the board members were chemical carcinogen people. So this used to be an authority journal for chemical carcinogens, how they are metabolized, what is their mutagenic, those type of, nothing at a genetic level. Okay. So the only place we could publish was Nature Science PNAs. And we used once in a while go to cancer research, and very often our papers were not well received there because that's not their major emphasis.
Maybe the opponents of you, because they don't want you.
So I decided to start, Elsevier came to me and said, we will start will you be willing to start a journal on oncology based on your handbook? We published it. At the same time, a friend of mine, in London, John Jenkins, was approached by Macmillan Publishers to start a journal. So I happened to visit London on a symposium, and John Jenkins came on a sabbatical to Hoffman LaRoche. Then so I had somewhat of not a great experience with Elsevier in publishing the handbook. I wanted this to be, available to students at a very cheap price. And they were not willing to put out a cheaper version of the, paper
publications.
cover. They put the price at $350 a book, which is completely out of for a student.
At least for India, where we come from, right?
Yeah, certainly in India. Even in US, no student can afford to pay $350 for a book. So it became essentially a library journal. You had to go to a library to read this book. And, I was not too happy with that. And I was negotiating what should be the subscription price of a journal, stuff like that. And Macmillan were much more receptive to having a very cheap subscription price for individuals. And, they were also publishing Nature. They were already a very profitable company. And, and when John Jenkins and I sat together, we felt, why create two competing journals? We can put together one journal. He can manage London, Europe, and all European countries' papers, and I can manage all American papers. And, and that's how we started. And we've started a journal. I was the editor for 25 years or something like that.
I have no clue how you manage everything.
And so that's the thing. The journal became so big, it became a weekly issue, and I was getting like 300 to 400 papers a month to review. So it became a little too hectic, and Macmillan wanted to change editors because they don't want the same people running the journal, which was fine. And then I started another journal called Genes and Cancer. It's more, I wanted it as a review-ish journal. And so where you give comprehensive reviews of the most modern topics, and that's what we're doing now.
That's fantastic, fantastic. So I mean, maybe last one of the questions, like you have seen the field, at least the cancer field was in the cloning and all the things, they were underdeveloped or not developed at all. And now you are seeing fast forward where the PCR can be done in 10 minutes, 15 minutes. And you
Yeah.
can get a you can get a sequence in 1 hour or so. Which was kind of impossible to think at that time, right? Now
Yeah.
with this kind of things that you have seen, where is the future going on? What do you think? And where how the people
I
have
think,
to prepare? Yeah.
yeah, I think we still, people think there are a lot of targeted therapies and all that. I think that field is not fully developed. You really need, I mean, this is a great opportunity for India. They have extraordinarily good chemists here. And, they're capable of synthesizing new molecules. And also Indian medicinal chemistry is very different from Western medicinal chemistry, because Indian chemists are used to extracting compounds, small molecules from plants and making derivatives of these things. So, I mean, when you do that, they tend to be much less toxic than, than what normally synthesized. And so there is a great opportunity in terms of developing new molecules for all these already identified targets. You don't have to go and identify a brand new target. We already know what are all the targets that work. And one can develop better and safer drugs using the power of chemistry in India.
Okay. And combination with, let's say, our own age-old Ayurvedic medicine.
Yeah,
because that's where all the
it doesn't have
medicine
to be in
plant.
combination. The Ayurvedic medicine works and at the same does not work, mainly because these plants, just give you the example of Taxol. Okay, this was discovered by an Indian medicinal chemist, Mansukhwani. Okay, Mansukhwani is from North India. He comes from a small village. He went to Bombay University to study chemistry. And when he joined NIH as a chemist, and he went to visit his mother in a village, and they were living in a small home with a yard. All the border is not a wall, but these vinca plants. Okay.
Yeah. Okay.
These purple flowers, beautiful flowers.
Yeah. Yes. Yes. Yeah.
So he asked his mother, why do you guys plant these vinca plants? There are so many other nice flowering trees. Every house has a vinca plant. He said, there's only one reason. Goats don't eat vinca plants because leaves are highly toxic. And he realized that's his start of his research career. Vinca plants make vincristine, which is a cancer therapeutic agent. So he started looking for plants which are not eaten by goats.
Okay.
So he isolated Taxol from yew tree barks, because you give these barks, yew tree barks, no animal touches it.
Wow. Okay.
So he discovered etoposide, he discovered Taxol, and 30% of cancer patients are today treated with these two drugs discovered by him from Indian plants.
Wow. And look at the wisdom and the way how he compared with what animals are not eaten. And the thought came from his mother or somebody who knows this stuff somehow.
So going to Ayurvedic medicine, the reason it is not taken over the world is because the activity is low. Ayurvedic medicines don't cure things dramatically.
Yes.
Because these plants are not making this to cure your disease, it's for their own purposes, for whatever it is.
Yeah.
Okay. So you have to do some chemistry, enhance the activity, identify the compounds, how they work, and there is a whole field of research that can be unraveled if there are proper resources allocated to it. And you can
Hopefully,
develop a whole new class of drugs.
our Baba Ramdev does it because they have a big Ayurveda institute in the north for Patanjali.
Yeah.
Great, sir. Anything that you want to share? Maybe anything
I think
for
I
the
have
youth?
shared everything that I can share.
Thank you so much, sir, for your time. It's an extraordinary journey that you have taken, going from studying from Tirupati. Now you are sitting in one of the prestigious institutes in the world. And I mean, I was actually counting your career after PhD. Like you finished in 71. And then now we are in 2024. It's like 53 years that you are in this cancer field. It's fascinating.
Thank you.
Great. Thank you so much.